Hello, my name is Jaroslaw Harezlak, and I'm a professor of biostatistics at the School of Public Health at Indiana University, Bloomington. And today I will talk about the statistical analysis that was done in our manuscript, in our published paper, BUAN, Bundle Analytics. So let me start with a brief outline. So what I will start with, it's simple statistical analysis. Then I'll describe twists and turns that actually happened for majority of the data that we are interested in, for the questions that we are interested, scientific questions we're interested in answering. Then I'll go on with the linear mixed models, which are the primary tools that we're using in our work. And then I will talk about incorporating data that incorporating the data that lie along the tracts and the streamlines in the white matter. So I'll present some visual summaries, some testing, and I'll finish with conclusions and some open problems. So when we're usually interested in scientific questions, we might rely on simple statistical analysis. So for example, we might summarize extracted quantities, for example, fractional anisotropy along either streamlines or tracts. And some possible questions that we can ask are, is there a difference between these FA values between, let's say, patients and controls? Or another question might be, are there any associations between the fractional anisotropy with disease severity? Okay. So simplistically, we might answer such questions by associating the summaries that we created in the first point with either the binary outcome, which is an indicator, either patients or controls, or with a continuous outcome, which can be represented, like here in question 2, by cognitive ability. So for the simple analysis, simple statistical analysis, we can use linear regression. So if we have a continuous outcome Y and predictor X, we can fit like just a simple linear regression model. So something I'm sure everybody has seen. Now, if we have patients and controls, again, we can fit a simple statistical model. So trying to classify patients and controls, and we might just use one covariate for that. And then, as you can see here on the bottom left, we see just a simple box plot here of the values. And these are values from the mean diffusivity. But we can also represent that as in the plot on the right, when we might model probability of somebody being a case, denoted by 1 in blue, or somebody being a control. So we all wish life was simple, but unfortunately we have to deal with some twists and turns in the analysis. So for example, we might want to adjust for confounders, for example, age, sex, socioeconomic status, education, or many other things we might want to adjust for. This is usually not a big problem in the statistical analysis, and both a simple linear regression and logistic regression can incorporate such adjustments. However, what I will spend most of the time in this video and in general talking about today is dealing with more complex study designs. So for example, we can have repeated measurements or longitudinal data, cluster data, hierarchical data. How can we deal with that and still answer the primary questions that we might have here? So let me go briefly over these different types of measurements. So repeated measurements, well, we observe, let's say, the same individuals at multiple common time points. So let's say we might have a study where our study participants enroll in the study. So we take measurements at baseline, but then they come back at 3 months, at six months, and let's say at a year. So we'll have 4 repeated measurements for each individual, And we have to take that into account when we're analyzing the data. Similar cases with longitudinal data, again, we make measurements on the same subject, same individuals, but they can be measured at multiple random time points. So by study design, they might not show up at the regular time points, but those time points might be driven, let's say, by the disease status. Now with cluster data, well, we have measurements, again, on individuals that come, for example, from the same families. This is the case, let's say, in Human Connectome project, where we have a lot of data that came from siblings, from twins, and these are both monozygotic twins and dizygotic twins. And again, in order to properly analyze such data, we have to take this data structure into account. And kind of all-encompassing data structure is this hierarchical data, where that contains all the other data structures that we discussed so far. So how can we model such data? It would be great if there was just one class of models that could take into account all those twists and turns. And actually, linear mixed models are almost a perfect tool for such data. So we'll describe them briefly more from the theoretical point of view first, and then I will describe how we used linear mixed models to actually analyze the data from the two groups of individuals, from patients and controls, and while we took into account the study design in the analysis. So let me go through a quick introduction in the next few slides. So what are linear mixed models? Well, here, as I said, it's a bit of a notation, but basically we have an outcome y, we denote outcome yi, and then we have predictors x. So this might be, say, group membership, patients versus controls. But now, in this matrix Z, we actually include covariates that are associated with so-called random effects U. What are those random effects? Well, those random effects actually will help us to take into account the study design. So we'll be able to deal with repeated measures, with longitudinal data and cluster data, because those random effects will induce correlations between observations that we obtain from the same individual. So, as I mentioned on the last slide, those coefficients beta, they are so-called fixed effects, and they describe the kind of the group effects or the population effects. So as I mentioned, we might describe in those fixed effects the group differences between, say, patients and controls. Random effects or subject-specific effects, they will help us to incorporate all the correlations that we have in the data. So here, this could be either, as I said, subject-specific effects or track-specific effects. And then those random effects will allow us to actually account for the correlations that arise from data coming from the same subjects, from the same tracts, let's say the same family structure. So here, as one of the examples, when we used such modeling strategy, here we had 3 groups for whom we're modeling the data. And here, many participants in the study had either 2, 3, or 4 observations. So these linear mixed models allowed us to model group-specific trajectories over time, over these 4 time points. And what we have here on the y-axis is mean diffusivity, and on the x-axis, we have the observation time points. So here, the correlation was taken into account, and the analysis here showed us that actually there were differences between all 3 groups, well, between the red group and the other 2 groups at baseline, but then those differences were diminishing over time.